Hello, I'm Bretton, one of our MCAT tutors here at Inspira Advantage, where we help students get into med school and other professional programs. Today, we're going to talk about all of the different characteristics of viruses that you need to know to do well on the MCAT. Let's jump on in. Let's start with the anatomy, specific the capsid. The capsid is a protein coat that encloses the viral RNA or DNA. The capsid is made of several repeating structure subunits called protomers. Nearly all viruses have a capsid. The next part of anatomy we want to know is called an envelope. The envelope is the outermost layer of a virus that protects the genetic material inside the capsid. The envelope is most important for the virus when traveling between host cells. And again, not all viruses have an envelope. Viruses may also have a sheath which connects the base plate and the capsid. The base plate is actually what binds to and infects cells. It has little pins on the bottom and tail fibers which help it grab the outside of the cell. Not all viruses have a base plate or tail fibers, though. You have all of the viral anatomy you need for the MCAT. Next, let's talk about the different kinds of viruses. The first type, and roughly what I drew here, is called a bacteriophage. These are viruses that infect bacteria. They have a tail sheath that injects DNA/RNA into the bacteria and cause its infection. There are also viruses called vibroids that are small RNA viruses that infect plants. The rest of the classification of viruses can get complicated, so I'll go through this carefully. Viruses can either be positive or negative sense. A positive sense virus can be directly translated into proteins by the cell, whereas a negative sense RNA virus must synthesize the complementary strand, which then can be turned into proteins. And it should be mentioned that it is the host cell that is doing this translation into proteins. The next type of virus we want to look at is called a retrovirus. And this isn't a virus from the 70s. It's a single-stranded RNA virus that uses an enzyme called reverse transcriptase to turn its RNA into DNA, then inserts itself into the host genome. So let's zoom in and see what this looks like. So we start out as RNA, which turns into DNA that can then insert into the host genome. So imagine in purple here, this is the host genome, and we've just inserted a piece of the yellow retrovirus DNA. So it'll be transcribed and translated as long as the host DNA is. HIV is a really popular example of a retrovirus. There are also two important life cycles a virus may have, the lytic or lysogenic stage. The lysogenic stage is when a virus integrates itself into a genome as a provirus or prophage. There, it will lay dormant until something like stress activates it. Typically, this activation will cause the lysogenic phase to enter its lytic phase. These viruses are really ready to kick you while you're down. So what is the lytic stage? Well, the lytic stage is when the virus replicates like crazy inside the cell until there's so much virus that the host cell explodes or lyses. Once you have this information down, you'll be ready to answer any virus question the MCAT will throw at you. Good luck. Thank you for watching our video on viruses, and I'll see you next time.